Hi everyone and welcome to Asami Rat Care. So today's video I'm going to be talking a little bit about genetics, and this is one that I put off for a while because it's one of those topics that scare the hell out of some people, in part because they are a very technical and complicated subject if you just kind of like look at them in their entirety, and it's something that I'm by no means an expert on. I stopped doing biology with GCSE, which is when you're about 16 if you're outside of the UK, though some of my friends did carry on doing it. And I did do their genetics paper based off my rat knowledge and did quite well at it just on their mock, which was quite funny. But it's something that it doesn't need to be hugely complicated, and that's why I'm doing this video really. I'm trying to kind of like explain some of the language, the terminology used, and then go into the logic, because actually it's a very logical subject. Some things are simple and some things are more complicated, and I'm going to try and stick at first to the kind of simpler kind of side of things. But what I will say is, if you are a geneticist or have kind of done genetics to a high level and are familiar with the terminology and such, one, there's no point watching this because I'm not going to teach you anything, but two, bear with me if you do decide to watch it, because I'm not going to use things properly. I'm probably going to tread all over technical terms. I tend to describe things in a way that makes sense to me, not necessarily in a way that is kind of scientifically perfectly accurate. But in terms of breeding, you don't need that scientifically perfectly accurate to understand what's going on. So that's really what this is about. It's about kind of introducing you to the basics of genetics, and I will follow this up with more. I have already filmed a copy of this that was 50 minutes long and decided that that is too much for anybody to take. So I decided to break it up further into kind of smaller chunks and try not and do too much at once, because it is a topic that will put a few people off if I do. Right, so without further ado, let's get on with it. So what are genetics? Genetics are basically the information that allows the body to form, and this is based off some information we take from our mum and some information we take from our dads, and that comes together and that creates the instruction manual that describes how to build us, for instance, be that us as a human or a rat or so on. It has all the information needed to build it. Think of it as like the world's best IKEA kind of instruction manual, probably a lot simpler to follow than some of the IKEA ones. But effectively that's it. It's kind of a massive kind of manual full of lots and lots of information, and it's not just describing what colour our hair is, even though this is genuinely not the result of genetics, or what colour our eyes are or what position our ears are in, etc. It also describes how to build skin, how to build hearts and lungs, and how effective that's going to be, and to a certain extent our personality, our health, our long-term health, and so on. So there's loads of information in this, and actually the stuff that we can see, that we can kind of look at a particular animal and say, right, you are going to be an agouti rat, or you're going to be a brown-haired person, or you're going to have blue eyes, it's actually very small compared to this massive manual that goes through loads of depths, and that we only kind of really shave the surface off in terms of our understanding, unless you're somebody that kind of dedicates your life to it. So it's a fascinating subject, very deep, and it's something that I'd like to go into more depth on some of the hidden aspects at a different point. But for now we're going to focus on the stuff that we can see, the kind of physical varieties of the rats. So thinking about that, we're looking at a very small section of the book, and what I will mention briefly is it's not like you would just look at a chapter. So all the information we're thinking of that look into kind of what eye colour a rat might have, what hair colour a rat might have, what ear position, how curly its coat is, that is not all kept in exactly the same part of the book just to be complicated. It's dotted around all over the place. And what you might hear people throwing around is things like DNA. DNA is effectively a language that the book's written in. It's how it's written, the words, etc. that make up the sentences. From our perspective, when we're trying to work out what's going on with our rats, it's not hugely important. It's just something that's going on that's kind of complicated and very, very effective at communicating, but doesn't affect how we see our rats at the kind of level that I'm dealing with. So effectively we're looking at this, and each of these kind of like genetic manuals has got various different sections in there, but what is common is they have a lot of pages, and those pages we call locuses, loci if you wanted the plural and be fancy, which is basically a posh way of saying a location. That's just it. You flick through the book, you pick a page, that's your location. That tells you everything that's going on with that particular gene, and there are loads, millions. I probably should kind of come up with an actual number, but I suck at remembering numbers and it's not particularly useful. But there are absolutely loads of these locations, and they describe all sorts of things, and that basically, the locations that we're interested in will tell us a particular fact about an animal. It will show a particular thing. So for instance, brown eyes and blue eyes in humans, that is one of the most simple kind of simplistic ways of looking at an eye. Rats. Humans' eyes can be brown or they can be blue. They can be different colours, obviously green, hazel, etc. Within that, that's kind of affected by different things. But if you look at it from a very simplistic point of view, kind of switches between those two things, and that is all defined at a particular locus, a bit like the most important locus, in my opinion, in a rat, which is the agouti locus, which tells you whether a rat is going to be agouti or not agouti. It's very simple, very straightforward locus, and I will go into it in more depth in a future video. At this stage in the last video I got hugely carried away and spent about 50 minutes talking about it, but effectively that's all it does, it's a switch. So a little bit of a diagram. This pen is a little bit rubbish because one of the rats ate it earlier. I will say it's non-toxic, but it is a little bit worse for wear. So effectively we've got a switch, and we either have on or off, and that describes the vast majority of the locuses we're going to deal with in rats. Most of them have an on or an off position. There's a few that are a bit more complicated that have kind of on, off, partly on, and there's a few that have on, something different, something different, something different, something different, something different, off. And that kind of, those get quite complicated, and we're not going to look at those right now, but I do want to kind of let you know that things do get more complicated, but the majority of things that you'll need to understand are fairly simple on off. It depends what variety you're choosing to breed or you're particularly interested in looking into. I just want to kind of concentrate on simple at the moment, and we'll get onto more complicated stuff later, assuming that I managed to keep this one up. So on and off, and where that kind of switch sits really depends on what you inherit from your parents. So we know that all of us inherit some genes from our parents, some from our mum, some from our dad, and effectively at each of these locations we generally have two kind of little genes, and I'm going to grab a demonstration of this. Ignore the fact there's two different types of balls, I was just going for the same colours. So effectively we have, each of us has kind of two different slots in us, and this is a ball that I've inherited from my mum, and this is a ball I've inherited from my dad, and they're the two kind of little bits of the genes, and they both kind of look fairly similar little things, and they could define what my eye colours are, what my hair colour is, etc. Anything that's a simple on-off type switch. And there are two different types. So when we're thinking about agouti, which I will use just because it is a very simple one, it's one I understand fairly well, we should hope, because I've been breeding agouti for 9 years now. But yes, so there are two different kind of options for agouti. There is the green, which we're going to say here is the dominant. These are quite important terms. You'll find them chucking around all the time, so it's worth memorising them. Not every term is that important. Locus is another one that I'd remember, just because that's another one that gets thrown around quite a lot. So we've got dominant and we've got recessive. And the thing with dominant is kind of like big, strong, it's heavier. So if we were to kind of put these on the switch, and we're saying green ball and a yellow ball is the recessive. So if we were to put these on the kind of seesaw, which is the switch, the dominant gene in this case would be heavier. So effectively it would weigh it down. If you've got a copy of each of the genes, you would have this would be heavier, so this would go in the way, and all you would see is the dominant gene. it might be carrying this, or actually it might be carrying this. It could be either. So all it kind of is making that difference is you're only seeing the dominant gene, because dominant is powerful. Dominant kind of like takes over, and all you see is that dominant. In the case of agouti, that means agouti. When you're talking about recessive, if you want to get a recessive to show, that won't work. It will always be hidden by the dominant gene. The only way you can see recessive is when you've got 2 copies of the recessive gene. Otherwise the dominant gene would just take over and hide it. So you can kind of get 2 copies of that. So if you had 2 copies of this, it would push it towards the kind of off in the case of agouti, or it really does depend on what that gene is. So the agouti gene, on is on the dominant side and off is on the recessive side. However, there's a lot of other genes out there that don't have that around. They're what we call a recessive gene. So actually their on is recessive and their off is dominant. And I wouldn't get hugely hung up about that at the moment. When we get into the individual kind of locuses, we'll talk about what the effect of the dominant kind of is and what the effect of the recessive is, because I will say most kind of varieties out there, like Russian blue, like mink, like the chocolate that we have in the UK in the main, there is an Australian chocolate that's determined to be different and is a completely different gene. They're all recessive on. They're recessive genes, so they need kind of 2 copies of the yellow ball to show up, whereas the dominant gene needs either 1 copy, or it can be one like that and it would still show up. So agouti and Rex, to an extent, we'll talk about Rex at a different point, they're dominant genes, so, or dominant chocolate, which is what we call the Australian chocolate that's knocking around. They only need 1 copy of this kind of like dominant gene to show up. So that's kind of dominant recessives. I would say with that, if it's kind of confusing the hell out of you, it'll make more sense if we do more examples, but I don't want to get into examples again, because then we'll have a 50-minute long video and everybody will cry, including me probably. So what I instead want to do is talk about how we inherit things from our parents. So I'm just going to get a pot, which is a plant pot, quite conveniently. So when we get our information from our parents, depending what our parents carry is what we inherit. So effectively, what we're going to do now is we say we're going to have a parent with 2 copies of the recessive genes. So every locus has 2 genes. That's just it. There can only be 2 genes sat at that location. That's the only thing that can fit effectively. So if we've got a rat that's kind of like a not agouti or a self or a black rat, then we've just got those 2 options in there. So what I do when the baby comes along is I randomly put my hand in. I can't see what I'm picking up from here. Weirdly enough, because there's only 2 options that are recessive, I picked up one of the recessives. Now, if I had a parent that was had 1 copy of the recessive gene and 1 copy of the dominant gene, so it would look like the dominant gene, in this case, let's say agouti, but it would carry the recessive gene, which in this case is black or not agouti, as we would probably call it, because that's how the switch works. It's either on or it's off in this case. So we would have those 2 options, put them in. When I was trying to think about what kind of gene it would inherit from its parents, I would just put my hand in and pull one out. It's picked up the recessive again, but actually if I repeat this multiple times, this time I picked up the dominant, this time I picked up the recessive, dominant, recessive. Weirdly, it's working out 50-50. Sometimes it doesn't work out like that. But every time we have that kind of mating incident occur in conception, we have that kind of random thing. So as well as looking in this bucket, which might be the dad bucket, we'd have another bucket at the same time, which would be the mum bucket, and we'd be making the same choice. In this case, we've got a rat that's kind of part dominant, part recessive gene. So with the mum, we're picking out a dominant, recessive, dominant. It's quite amusing that it's kind of every other one. I must be kind of subconsciously picking it somewhere. But effectively that's it. It's a lottery. We put the balls in there, we muck around and pick one out, and that will then define whether we inherit recessive or dominant from our parents, if the parents carry recessive and dominant. And this is a kind of another term that's useful. I don't particularly like it because it's long and I can't spell it, which is anything that's long really. There's two terms here, which is heterogeneous and homogeneous. So homogeneous has two copies of the same thing. So whether that is two yellows or two recessives, or two greens and two dominants, they are homo, so they have two of the same thing. A bit like you'd have homosexual, two of the same sex partner. That's how I remember it because I've got a crap memory. Heterogeneous is basically you've got one copy of dominant, one copy recessive, one copy dominant, one copy recessive. Kind of wherever it happens around, it's hetero, they're two different, a bit like heterosexual, kind of partner of the different sex. So that's kind of like your two main options. You've either got parents that have exactly the same or parents that it's got different. So like I have brown eyes, so when we talked about this before, we kind of mentioned that brown eyes could be kind of switched between brown and blue. My mum has blue eyes, my dad has brown eyes, so we know that in the kind of great mix-up, my mum effectively had two copies of the recessive, so she has grey eyes. She must have. The only way we can kind of see her kind of grey blue eyes is her having two recessives. If she had a copy of the brown eyes and a copy of the blue eyes, the brown would overpower the blue, so she must have two copies. So we put that in the bucket. When the kind of great decision of what Gemma's going to be made of comes out, I have to pick out, and I'm always going to pick out a yellow ball because there are only yellow balls in there, so there's only recessive options. So I must carry grey eyes. Now, with my dad, we know he's got brown eyes. I don't know what he carries. He may have two copies of this, or actually he may have one of each and we can't see. And there is no way of telling that. So actually what we kind of do, and I'm going to pick a letter for this, we quite often describe things by letters, and I'll stop around about here before we get onto Punnett squares because I'd like to do a video entirely on Punnett squares just because they're useful. But we use a certain amount of kind of names for this. So when we're talking about green, and let's use A for this. It is the agouti locus symbol, so we use different letters for different locuses to try and describe it. It's just a way of kind of being able to write it down easily. But in this case, it could mean anything. So dominant would be a big letter because it's dominant and big. Recessive would be a little letter because it's recessive and going to be hidden by this. And you can kind of almost picture it a bit more when it's like that. So we know dominant, recessive. We know my dad has definitely got two genes. So we know he's dad, mum. Let's write mum's first. So we know mum's got two recessives. So she would be two little A's, or two yellow balls. That's my yellow ball. And we know that dad has at least one green ball, one dominant gene, because he has brown eyes. Now, I don't know what his other thing is, so another thing that's quite useful to know is this little dash. That means I don't know. You could put a question mark if you wanted. It's really just to communicate to yourself that you're not sure. So I know with my mum, like I did before, when we're picking out which balls to pick, we put the two yellows in there, and I would always get out a yellow ball for me. But for dad, we're either going to put in two green balls, in which case, weirdly enough, I will always pick a green ball out, or the dominant gene. So whatever happens, if there's two green balls, I would always be brown-eyed. I would always carry this recessive gene. Or in one case, it could actually be that dad is recessive kind of for brown eyes, so he also carries it. I put those two in, and you know what, a bit like before, I pick out, oh, green, that means I've got kind of brown eyes, or oh, I've got yellow, but I'm already holding this yellow ball from my mum, and actually then suddenly I've got blue eyes rather than brown eyes. Now all we can say is I definitely picked out a green ball or a dominant kind of gene, a brown eye from my dad. We don't know what else he carried. I can say that my brother also is brown-eyed, so we know that he also picked up a dominant gene or brown-eyed gene from my dad. But that's actually only two of us, and actually it could be that dad still carries this, and if mum and dad had continued having lots and lots of babies, eventually they might have popped one out. As much as popped one out, that sounds a little bit kind of disturbing. But effectively, they could have had a blue-eyed child. what we have is every single time we make this choice, every single time we put the balls in there is a 50% chance. Once I take that out, it doesn't mean that my brother is only left with the blue eye thing there, because actually, as much as I've only put 2 in there, as soon as I've kind of finished, it resets and it will go in again, so we've got another kind of choice of those 2 balls. You could imagine this as being infinitely full of green and yellow balls, and I just put my hand in each time and pick it out, but it doesn't change the overall kind of probability of picking it. Just me and my brother both had, if Dad did carry blue eye gene, the recessive gene, we both had a 50% chance of picking that out. And we'll talk in the next video a little bit about Punnett squares and how you can work out that percentages, and also how unreliable that percentage is. But that's basically it. So I just wanted to cover off that genetics were instruction manuals effectively and how to build us, that locuses were a particular location in that genetics, that the page in the instruction manual that described how to do a particular thing, like what colour your eyes were, what colour your particular kind of like is a kind of rat agouti or not agouti, agouti or self, agouti or black, that kind of thing. And they work in this kind of on-off switch, that you get recessive genes and dominant genes, where recessives are always going to be stronger and overpower a dominant gene if they have both of them. And everybody at these locuses has a slot for 2 genes to sit in, one from your dad, one from your mum, or in a rat's case, their dad and their mum. And they could be filled with recessive or dominant. If a rat or a human or such has a dominant gene sat in there, you will always see the dominant gene over any recessive gene. Things do get a little bit more complicated when it isn't a straightforward dominant or recessive. You can get partially dominant, or you can get multiple steps, but we'll go into those when we look at specific locuses. For the majority, dominant, recessive, on, off, kind of switch position. And this idea that we kind of like pick from our parents. So when I say pick, we don't get to choose. It's a random selection, and you don't know what you're going to get for each mating from each of the parents if they carry 2 different things. And then also we're looking at homogeneous and heterogeneous. Homo, 2 copies of the same thing, be that 2 copies of the recessive or 2 copies of the dominant gene. And heterogeneous, you've got 1 of each. And that we actually can't tell necessarily if you've got somebody expressing the dominant gene like me, unless you know that my mum had the recessive gene, so I must have got one of the recessives off her. You can't always tell what they carry unless I had children and my husband happens to have blue eyes, and then we would be able to see that there's blue-eyed children born from that, in theory. Again, it's all probability. So that's the basics really, that's some basic words and language for you. I will try and film one on Punnett squares, which is what sounds horribly scary, but is actually just a really simple way of trying to understand what's going on at each locus, and then we'll kind of think about how things can stack up. And then what I will try and do is a video on some of the locuses. I won't say that I'll do all of them, because I probably lose the will to live after a while, but I will definitely focus on some of the main ones, and I'll probably particularly talk around the varieties that I breed. But I'll also talk about some examples of more complicated things as well, but not for a while, and I'll try and do separate videos for all of them. So hopefully that's okay. It's definitely substantially shorter than my last video that I filmed, and hopefully this is fairly clear, but Fu managed to chew through the wire on my my microphone earlier, so I've not got the microphone on right now. So over and out from me, and I hope that was useful.